Hello, my name is Gloria Robert. Today, we are discussing the topic genetics. Objectives of lesson. During the course of the lesson, the student should be able to 1. Define heredity. 2. Mention at least 5 transmittable characters in plants and animals each. 3. Define all the genetic concepts mentioned here and even more. 4. Highlight Mendel's work and state the laws of inheritance. 5. Diagrammatically cross genes and interpret them. And 6. Define incomplete dominance, codominance, and give their examples. What is genetics? Genetics is the branch of biology that deals with the study of variation and heredity in living organisms. Heredity is also known as inheritance. Variation has to do with the differences that exist between two or more organisms of the same species or population. Inheritance has to do with the transmission of similar traits within organisms of the same species or population. Now in Genetics 1, we studied variation. So in Genetics 2, we are going to be discussing inheritance. What is inheritance? Well, let me say, what is heredity? Heredity is the transmission and expression of traits or characters from parent organisms to their offsprings in a given population. Offsprings are the new individuals, or let me put it as children produced by parent organisms. That means we all are offsprings of our parents. This can also take place in plants. If you plant a seed, it germinates and grows into new plants. Those new plants are the offsprings of the parent plant. Now the characters expressed by these offsprings are inherited from parents' gene. Now study this image. We have parents and their children in this image. Now if you observe these children, you notice that they have certain characteristics, or should I say features, that are similar to that of their parents. For example, color of their skins, shape of their nose, probably color of their heads. All these are inherited from their parents, or they were transmitted from the parents to the children. So that is what heredity is all about. Now let's look at some of these transmittable characters. Note that only the characters or traits controlled by genes are transmitted from parent organism to their offspring during fertilization. That means environmentally acquired genes cannot be inherited or transmitted. What do I mean by this? There are certain characters that are acquired in the environment. For example, someone that gets blinded due to an accident, someone that acquired big muscles due to exercise, or let's say someone that bleached his or her skin. All these are environmentally acquired traits. They are not inside the gene of such a person, and so the children of such persons cannot inherit, neither can they transmit it to them. Now some of these transmittable characters are 1. Color. It can be color of the skin, color of the eyes, color of the hair, as in animals, or it can be color of fruits, flowers, seeds, and leaves, as in plants. Number 2, shape. Shape of the nose, shape of the head, shape of the ear, shape of the eye, shape of the mouth, as found in animals, or shape of fruits, flowers, leaves, and so on, even seed, as found in plants. Now number 3, here we have intelligence. Yes, intelligence too can be inherited by children or offsprings from their parents. Number 4, we have fingerprints. 5, blood group. 6, height. Height can be either tallness or shortness. Some parents are short, so their children too are short. Some are tall, and their offspring too are tall. Number 7, sickle cell anemia. 8, ability to roll the tongue. 9, voice or speech texture. 10, baldness. 11, albinism. And 12 here, color blindness. We have other characters that can be transmitted. So how is this done, or how are characters transmitted? Very easy. This process occurs during sexual reproduction. Now all the cells in the body of a parent organism, or all the cells in the body of an organism, apart from the gametes or the sex cells, they are all diploid. Now this means that they have 2 sets of chromosomes for a character, and so they are represented by 2n. Now the male parent produces the sperm cell, while the female parent produces the egg cell, also known as ovum. Now in plants, the male parent produces the pollen grains, while the female parent produces the ovum. So plants too can carry out sexual reproduction. Now, the sperm cell and the egg cells are called matured sex cells or gametes. They are involved in fertilization and they are haploid, meaning one set of chromosomes for a character. Now, characters are transmitted during fertilization by what we call meiotic cell division, or you can simply call it meiosis. Now, when there is crossing over of the male and the female chromosome, you see that this can occur. Now, what do we mean by crossing over? During fertilization, the male gene crosses to the female crosses to the male, thereby causing a blending of genes. And that is why you see offsprings looking like both the mom and the father, or both the male parent and the female parents. Now here we have this image of a parent. They gave birth to children, and so these children acquired some of the traits we are talking about all this while. Now, how does it actually happen? The male produces the sperm cell, the female produces the ovum, and inside these gametes we have the genes. So the genes of the male, they are found inside the sperm cell. The genes of the female, they are found inside the ovum. And during sexual reproduction, fertilization takes place. They fuse together and form zygote. The zygote develops into a new offspring or baby or individual. So by that time, such a baby has acquired the genes from the parents. Now let's talk about some concepts common in heredity. Number one, we have traits, also known as characters. These are the attributes or features inherited and expressed in an offspring from a parent. For example, height is a trait. Color, maybe color of the skin, color of the hair, they are all traits, they are all characters. Even tallness, like I said, height, color, shape, and so on. All these are traits that can be inherited. Number two, we have genes. Genes are units of inheritance that controls inherited characters, and genes are found on chromosomes. Another one we have to know about is what we call alleles, or you can call it allelomorphs. These are pairs of genes that control contrasting characters. For example, tall genes and short genes are contrasting characters for a character, which contrasting genes for a character height. For example, you have height. Height can be expressed in different ways, either as tallness or shortness. You can even have color as a character, and it can be expressed in different contrasting ways, such as red or blue, white or pink, and so on. Now, another one we need to discuss is phenotypes. These are the sum total of the traits expressed in an organism. They are usually dominant and are influenced by environment and genotype. What am I trying to say? Now, if you observe an individual, all the characters that are expressed in that individual, you can see those characters are phenotypes. For example, you can see that an individual is tall, an individual is fair in complexion, an individual has a starry kind of eye. All these are physically expressed genes. Some of them might not be physical, but you can see them, such as intelligence and so on. So far it is expressed in an organism, it is known as a phenotype, and it is dominant because that is what you can see. Now, next one is genotypes. These are the sum total of all the genes acquired by an offspring from their parents. Now, all the genes an individual acquires from his or her parents make up the genotype of such a person. It is also the total genetic makeup of an individual. What are the things that can influence genotype? Yes, parent genotype determines your genotype. Don't forget, you acquired your genes from your parents. And of course, mutation. Mutation is a sudden change in the genetic component of an individual, probably due to exposure to certain maybe radiative materials and other things like that. Okay, it could even be exposure to X-ray and the causing a change in the genetic makeup of an individual, and so it can change the phenotype and of course the genotype of an individual. Okay, let's look at dominant traits. These are the traits that are expressed in an organism. Okay, they are expressed in an organism and they are represented with a capital alphabet letter. So when you see capital T, capital R, capital S, anything alphabet written in capital letter, such trait is representing a dominant trait. They are controlled by dominant gene. We'll talk about them more. The next one is recessive traits. These are traits that are not expressed in an organism, even though they are there, but they are hidden or masked. Someone that is dark skinned, yes, that dark skin is the dominant trait. That is what people are saying. But inside the gene of such person also have recessive gene for fairness. So that person is carrying the gene of fairness, but it is hidden or masked. They are represented with a small alphabetic letter, okay, alphabet letter, such as small letter T, R, S, and so on. So they are controlled by what we call recessive genes. Now, homozygous. These are the traits in individuals with two sets of similar genes for a character. For example, you can have character height, and you want to talk about tallness. Tallness can be capital TT, meaning that that person is homozygote tall. It can be small letter TT, meaning the person is homozygote short. It can be capital letter SS, homozygote smooth, or small letter RR, homozygote rough. Okay, we'll talk more about that. Next, we have heterozygous. These are traits in individuals with two sets of different genes for a character. So you have two different genes. One is a capital letter and the other is a small letter. The capital letter is the dominant gene. The small letter is the recessive gene. So you have examples here for you to go through. However, note, no two different alphabets can be used for a trait. For example, we are talking about tallness and shortness for height. Tall, you use capital T. You cannot use S to represent shortness. Rather, you use small letter T to represent shortness. Or you are talking about smoothness and roughness. Okay, you use S to represent smooth, and then you use small letter S to represent rough. So you can't use two different alphabets. Take note. Okay, let's continue. Now let's look at Mendel's work. Gregor Mendel worked on heredity. How did he do this? He used what we call the garden pea, known as Pisum sativa. Why? Because they have the ability to self-pollinate themselves, they have short lifespan, and they have several unique features such as different shapes of seed, different colors of flowers, and so on. So he used the principle of hybridization. So what is hybridization? Hybridization is the crossing of two contrasting characters to get a different or a unique one in an offspring. For example, you have a tall person or a tall seed and a short one, and then you cross them together. You can get an offspring that is not as tall as the father or the male and not as short as the female. So very unique, different from both parents. And these offsprings are called hybrid. For plants, we call them hybrid. Okay. Now, there are two types of hybridization, which are mono-hybridization and dihybridization. So we are going to be studying them one after another. So let's start with mono-hybridization. Now this is the crossing of two pure hybrids. Okay, you can cross capital TT, that is pure hybrid. You can cross small letter TT, that is another pure hybrid. Or we can simply say it is the crossing of a single character with contrasting gene. For example, crossing of height. Don't forget that we can have height as in tallness, height as in shortness. So when such is crossed, only one character is mentioned or discussed or crossed, such is monohybridization. So what did he do? Number one step, what he did was he crossed homozygote tall plants, homozygote, the same gene, capital T, so they were tall, right? And all the offsprings or children got it, they were all tall. Then he crossed homozygous short plant, and all the children or offsprings were all short. But don't forget, we are discussing seed here, so use offsprings or seeds. All right. Now step 2, he crossed homozygote tall plants, capital TT, and then crossed with a short homozygote plant, and then all these children or all the offsprings were all heterozygote tall. Hetero means different gene, and the different genes must not be in two different alphabet letter, must be in the same. One is capital and one is small. So this is how to write heterozygous tall. And here is the crossing of what he did in step 2. He crossed a parent that is pure breed homozygote tall and a parent that is pure breed homozygote short. And then in the process of crossing, you have to separate them to show that they are gametes, right? And what I do to help me to cross, I number them. The first two, I call them 1, 2. the second two I number it 1, 2, and then I use my unique formula. What I do is 1 on this side, I fix it to the 1 on the other side, 1 on this side, I fix it to the 2 on the other side. Then I move to the other side, 1 here, I fix it to the previous 2, then 2 here, I fix it to the 2 from the first part. Just go through it, you see how, though we discuss it better. And now take note that we have what we call first filial generation. Now first filial generation are like the offsprings. So the crossing, the first set of offsprings produced are the first filial generation. You can simply call it F1. Step 3, he crossed 2 of F1, that is the first set of offsprings. You just pick 2 and cross them together, that is self-pollination. Now the next offsprings were in ratio 3:1, meaning 3 were tall and 1 was short. Okay, F2 means the next set of offspring produced from the previous offsprings. For example, I am a parent, my children are my first filial generation, so my second or my grandchildren are the second filial generation. I want to believe you get. Yes, you get. Like I said, I have my unique formula for crossing, though there's a video on how to cross gene. Just take time and look for it. Now what I do is I number it 1, 2 on the right side, 1, 2 again on the left hand side. Then I pick 1 from the right, join it to 1 from the left, 1 from the right, join it to 2 on the left. Then I move to the left side, 1 from the left, I join it to 2 on the right, and then 2 on the left to 2 on the right. So just follow this, you should be able to cross neatly and very interesting. Don't forget to write parents, gametes, crossing, and of course first or second filial generation. Take note, you must be able to differentiate between phenotype and genotype. Phenotype means what is expressed, probably what you can see physically. That means 3 of the seeds were tall and 1 was short, and that is ratio 3:1. Genotype means you can't see them, so it is the gene, either they are heterozygous or homozygous. So let's go through the second filial generation. We have 1 homozygous tall. Don't forget that capital letter means that is what you can see, what is expressed. So the 2 at the middle, they are 2 heterozygote tall, and the last small letter tt are the 1 homozygote short. So, and that is in ratio 1 to 3. Good. Now Mendel's first law of inheritance. This law is also called law of segregation of genes. This is actually as a result of his work on monohybridization. And so the law states that hereditary characters of an organism are controlled by genes. We already know that genes controls characters that are inherited, and that they are transmitted from one generation to another without alteration from other genes. For example, if you have 2 genes for a character like tallness and shortness, these are genes, right, for a character. Now when they are being transmitted from parents to their offspring, they are being transmitted individually. They move from parents to the offsprings individually without any alteration from one another. So gene tallness does not disturb the transmission of gene shortness. Each of them are transmitted from one generation to another without alteration. So I will come again. Now this law of segregation of genes states that hereditary characters of an individual or organism are controlled by genes and are transmitted from one generation to another without alteration from other genes. Take note. Now let's look at the hybridization. Now this is the crossing of 2 different contrasting characters in seed. In the first one, monohybrid. Mono means 1. We talked about a single character. In this dihybridization, di means 2, and so we are talking about 2 different characters. Here we have color and shape, and color can be contrasting as well. So we have yellow and green, shape can be round and wrinkled, and so on. So now at the end of all the work he did on this dihybridization, he came up with offsprings which are in ratio 9:3:3:1. 9:3:3:1. And the meaning of this is that he had 9 yellow and round seed, 3 yellow and wrinkled seed, 3 round and green seed, and 1 wrinkled and green seed. So this is the crossing of how it was done, the first filial generation, and the second filial generation was what produced this 9:3:3:1. And altogether we have 16 offsprings. Okay, don't worry, we're going to do a video on that, all right, for you to understand how this was done. Okay, so let's move on. Mendel's second law of inheritance. law of independent assortment of genes. This is the second law. Now, what is this law saying? The law states that characters are assorted independently and transmitted separately from one generation to another without alteration from other characters. What this is simply saying is that if you have two characters in a parent and these characters are to be transmitted to the offsprings, each of them will be transmitted independently without alteration from one another or kind of interference from one another. For example, character yellow, character, we are talking about shape now, roundness. These are two different characters. They will be sorted out independently, and then each of them will be transmitted to the offsprings independently without any alteration from one another. I believe you get. Yes, I know you've gotten me. Let's move on. Okay. Now let's go at test cross and back cross. Now back cross, this is the crossing of a homozygote parent with a recessive parent. Okay. Yes. Homozygote dominant parent and homozygote recessive parent. That is what backcross is all about, and you can see the crossing of that directly in front of that statement. And at the end, all the offsprings in F1, they were all what? Yes, they have the capital R. So let's take that one for redness. They were all red. Good. Now test cross. Now this is the crossing of heterozygote offspring. That means one of the offspring from F1 is crossed with a homozygote recessive parent. Okay, and then the F2, we have one homozygote red. Yes, we have two heterozygotes red, and then we have one white. So it shows what the same one we talked about in monohybridization, and so that is 1:2:1 for genotype. The phenotype is 3:1. Okay, let's move on. Yes, we've come to incomplete dominance. Incomplete, yes. This occurs when two pure breed parents, definitely they are homozygotes, okay, with two different genes, okay, two contrasting genes. Probably one is tall, one is short for height, okay, and then they interact or blend together to produce an heterozygote offspring that is different from both parents' phenotype. Very easy to understand. Let me use this illustration. For example, you have a red flower and you have a white flower. Now, all things be equal, you're expecting that the children or the offspring, of this flower should be either red or white. But here we have a totally different color, which is color pink. So why? Because there is a blending or interaction of these two genes to bring about a totally different offspring. So like I said, it occurs when two parents, homozygote parents with contrasting character, are crossed together to give you a totally different heterozygote offspring. The offsprings must be heterozygote because it will carry the genes of both parents. One is dominant and one is recessive. Okay, now this occurs in 4 o'clock plants, Mirabilis jalapas, Alausian fir, and so on. Okay, let's talk about codominance. This occurs when two parents with two different genes interact to produce a heterozygote offspring in which both parental genes are dominant. For example, you have a parent with blood group A and you have another with blood group B. Now it is expected, or you would have thought, that one will be dominant, one will be what? Recessive, yes. But in this case, the two of them are dominant. The two of them will be expressed, fully expressed in the offspring. So AB is a good example of co-dominance. All right, I want to believe you've gotten everything I've explained. But if you have not, you know what to do. Go back to the video. Right, now we have our student activities. Yeah, I love this part. This is the time for you to actually know if you have gained something from this lesson or not. So 10 questions for one intelligent, wonderful student watching this video. Quickly, probably teacher watching this video. It's time for you to get to work. All right, this is the answers to all our students' activity. Like I always say, if you are not able to get it, go back to the video, watch it, download it if you have to, view it again and again until you got everything I've explained here. Yes, our target is always 100%, and I want to believe you got that 100%. Please like my videos. Thank you. So at this time, I'll say thank you once again for joining me. Thank you for learning. Bye.